Hello everybody. Today we are going to see about the post-transcriptional modifications that happens in the mRNA. Before we go into the topic, let's just have a basic overview. What is an mRNA? What is the post-transcriptional modification? So first we know that we have a cell and we have the nucleus within it, right? So here we know that we have the DNA which lies within the nucleus. We all very well know that all the information that is necessary for the body is present within the DNA, right? So this DNA will now get transcribed into an mRNA. So what does it mean? That is a transcription. So what does it mean? The information that is present in the DNA is being copied into the mRNA, right? So this is the transcription. So whatever the modification that happens in this mRNA post-transcription, after transcription, is known as the post-transcriptional modification. Fine. So you should know that this is the DNA and this is the mRNA, right? So this mRNA, it is in the immature form. So it is either you can call it as immature mRNA or you can call it as heterogeneous nuclear mRNA. Why is it called immature or why is it called heterogeneous nuclear? Without this post-transcriptional modification, which I am going to tell you in a short while, this mRNA will not be able to survive within inside the cytoplasm, because there are many exonucleases. Nuclease, anything which ends with the word ase, it is supposed to cut or splice away our product, right? So here our product is the immature mRNA. So without the post-transcriptional modification, it cannot withstand in the cytosol, and for many other reasons also post-transcriptional modification happens. So now let's see what is the post-transcriptional modification. Say for example, we have an mRNA and this is the 5' end of the mRNA and this is the 3' end of the mRNA. This is our heterogeneous nuclear mRNA or our immature mRNA. Fine. So here this modification happens in 3 steps. So first one is capping. So what does capping mean? Capping is nothing but it happens in the 5' portion. So 7-methyl guanosine. Is now added to this 5' end by a special 5' linkage. Fine. So this is what is capping. So what is the role of capping? Capping is nothing but it will be, as I said previously, it will prevent this mRNA from degradation from the exonucleases that is present within the cytosol. Then in the middle it undergoes splicing. So what does splicing mean? Splicing is nothing but this mRNA will contain introns and exons. Say for example, so in short, this is an intron and this is an intron and this is an exon. So the exon is the coding portion. It is that which contains the information which has to be coded and translated into the protein. This intron is the non-coding. We don't want that which is present in the intron to be coded. Fine. So in this immature mRNA, splicing also means you have to cut and throw it away. So the introns are being spliced and the exons are then kept beside each other so that the mRNA becomes mature and the proteins are and it is translated into a protein. This is called as splicing. Okay. Now the other one is the polyA tail addition. So that is the polyA tail, addition of polyA tail. A stands for adenine residues. So at the 3' end we add adenine residues to the 3' end. So this is the addition. So these are the post-transcriptional modification: capping, splicing and addition of polyA tail. So, once all these happens in the hnRNA or the immature mRNA, it becomes mature and it is able to survive in the cytoplasm and it gets translated, it gets attached with the ribosomes and it gets translated. Okay. So, let us see in detail about this capping. So, we all know previously that 7 by in the 5 prime 7 methyl guanosine is been added by a special 5 prime to 5 prime linkage. So, how does it happen? It happens by the 2 enzymes. So, it happens by 2 enzymes. First, the GTP is first added to the 5 prime end. Fine. This happens by the enzyme guanylyl transferase. So, we know that transferase is the enzyme which is used to transfer. So, guanylyl transferase transfers the GTP first. Then, we have our methyl group transfer. So, that is by means of guanine methyl transferase. So, with the help of guanine methyl transferase, which is the second step, we have this capping. Fine. So, how does splicing happen? Splicing happens by SNRPs. So, it is shortly known as SNRPs, but splicing happens by hnRNPs. That is nothing but small ribonucleoproteins. Small ribonucleoproteins are responsible for splicing. Okay, that is the removal of the introns and having all exons side by side. Now, what is a polyA tail? PolyA tail is quite interesting. PolyA tail, the addition of polyA residues it happens by the help of this enzyme which is known as the polyadenylate How does this complex add the adenine residues? This complex first identifies a sequence. That sequence is, say for example, that sequence is AA, it's AAA. So, this is the sequence. So, once this sequence is identified, polyadenate complex removes a part of the immature mRNA at the 3' end. Then it will add adenine residues to this 3' end. So, the adenine residues can be approximately about 25 to 250 adenine residues will be added to this 3' end by this enzyme polyadenylate polymerase complex by identifying this sequence. It is quite simple. First it has to have 2 AA, then U, then 3 AAs. So, this is the sequence. Once it identifies the sequence, it removes downstream to the sequence, it removes the immature mRNA, then it adds approximately about 25 to 250 adenine residues to the 3' end. This is in short about the mRNA, post-transcriptional modification of the mRNA.